Welcome to Teja Statistics. In this chapter, we are discussing the designs of experiments, which is our unit number 2. Yeah, designs of experiments is useful uh, for understanding the statistical data, and design of any experiment involves planning, obtaining relevant information regarding the statistical hypothesis under study and making statistical analysis of the data. For example, an experiment is conducted to examine the effect of three different drugs administered to five patients. To conduct this experiment effectively or efficiently, first the experimenter has to plan the major concerns like selection of suitable and appropriate patients which are relevant to the study, under which uh, climatic conditions the drug has to be administered, how much quantity of drug is to be applied, the better way of taking the measurements, etc. Proper care has to be taken to formulate statistical hypothesis to obtain relevant information to the study it. Eventually, an efficient statistical analysis has to be made, obtained in a valid inferences. This type of planning is called designs of experiments. So let's see the definition. The design of experiment may be defined as the logical construction of the experiment in which the degree of uncertainty with each with which the inference is drawn may be well defined. So what is an experiment? Terminology you have to consider here. Experiment means getting an answer to specified problem under certain conditions. So absolute experiments and comparative experiments. Absolute experiments are designed to determine the absolute values or precise values. For example, determining the average marks of a subject in a college, finding the correlation between the income and expenditure of the group of selected families. Next, comparative experiments or comparative studies are designed to the effect or two or more objectives of some population characteristics. A comparative study of three fertilizers on a crop. Here more than two variables are there. Analysis between different drugs made by a company, and study of different kinds of cultivation process. Treatment experimental unit and treatment, you have to know experimental unit and blocks yield the result. Okay, experimental error, this is a very important definition. Experimental error. Okay, just see, if we allocate the same treatments for all the experiment units of the homogeneous experimental material, but the yields still vary from experimental unit to unit, the variation is called the experimental error. Okay, the factors we have to find out. Okay, it is also known as noise. Okay, so you can see the example also. If we consider an agricultural experiment on which plots are homogeneous of equal shape and size, so if we apply the same treatments to all the plots, still the yield vary from the plot to plot. So this variation, such variation to the plot to plot due to random factors, soil fertilities, waters, is called experimental error. Okay, precision, very important definition. Precision, the reciprocal of the variance of the sample mean is known as the precision. Next, efficiency of a design. Efficiency of a design. Yeah. Next, we have uh, another definitions where we have to apply these designs of experiments. Designs of experiments is extensively used in agriculture, greenhouse studies, etc. The design of experiments is applied to study the laboratory techniques, medical experiment, pharmaceutical experiments, etc. Design of experiment is used in the field of economics and industry. Next, very important, this one, principles of an experimental design. These are the three uh, very important, which was given by R.A. Fisher: replication, randomization, local control. So what is replication? The repetition of an experiment. Replication means a repetition of treatments more than once under investigation, executing the experiment more than once. So the repetition of experiment, simply we can say that this is a replication. Replication is a process, so repeating the same treatment on various effects experimental units under similar conditions. Next, advantages of these replications. Replication is useful to reduce the experimental error. The large number of replications reduce the standard error, and it results in increasing the precision. Next, if you consider the second condition, that is randomization, you have disadvantages also. Okay, disadvantage: increasing the number of replications, it is not possible since it is involves more cost. Next, replication of treatments may be sometimes subjected to the bias. So next, large number of replications may be lead scarcity of the resources. Yes. Next, coming to the randomization, a process of allocating the treatments to the various experimental units in such a way that each and every experimental unit has an equal chance of receiving any of the treatments is called randomization. The main objective of randomization is validity of the statistical test of significance. All the statistical tests are based on that the observations and drawn randomly or independently. Next, the advantages for randomization. Uh, Randomization eliminates any kind of human bias. It provides logical bias for conducting various statistical tests of significance. Next, coming to the disadvantages, randomization is difficult to apply for of large number of experimental units. Randomization process can be effectively applied by the statisticians only. Next, coming to the local control. If the experimental material is homogeneous or heterogeneous, and different treatments are applied randomly over the entire material, if the experimental error is increased due to some uncontrolled factors, even the treatments are applied randomly in the sufficient number of times, then it is desirable to reduce the experimental error without increasing the number of replications and without interfering the principle of randomness. This is achieved by the principle of local control. Definition, just see the definition: the process of reducing the experimental error by dividing the relatively is called local control. A heterogeneous experimental material into homogeneous blocks or subgroups is known as local control. Yeah, just see the example. For example, in agricultural experimentation, heterogeneity is observed in various experimental units plots. That is, the experimental errors is increased. The experimental error is increased due to fertility, a gradient of soil, and even in different fertilizers have been applied randomly with sufficient replications. In addition to the principle of replication and randomization, the experimental error can be further reduced by the dividing or group of agricultural experiments into relatively homogeneous blocks. This is called the local control. So simply, if you are directly applying the homogeneous heterogeneous values, we can't able to get the exact information from the given data. But here we are getting the another uh, local control applications without randomization and without this homogeneous functionality. So we are just doing the heterogeneous plots into converting into some homogeneous subgroups. So then we will get easily the accurate values from the experiment. So that's why this local control is very useful. Local control reduces the experimental error. The local control ensures the experimental design is more efficient by reducing the experimental error to get a great extent. We can detect even a small differences between the treatments also. Our disadvantages also be there. If the experimental material is homogeneous, then dividing into the blocks is not necessary. Sometimes applications of local control principle is not much useful. Yes, in general, soil fertility does not follow any systematic pattern. Hence, the heterogeneous blocks is a difficult to task. Yes, these are the three definitions in principles of experimental design. You have to know, very important. Each and every time they are asking in our examination. Okay, so principles of experimental design. Next, before that we studied the designs of experiments, concept, introduction, definition and terminology. Is that okay? So in next class we are going to discuss so many things in designs of experiments. Thank you.